Biotechnology, the use of biological organisms and processes to provide useful products in industry and medicine. It has begun to affect so many aspects of our 21st century lives. Biotechnology applications can be seen in agriculture, pharmaceutical development, chemical engineering, and even the human genome. The use of this technology expands and grows each day. But what exactly is biotechnology? How does it affect everyday life? Where will it take us in the future? Biotechnology in the 21st century will help demystify this seemingly limitless and vital science by providing concrete answers to these important questions. Life on planet Earth all started with a bang. From a single point of compressed matter to the vast and infinite depths of space and time, millions of years of evolution and growth have led scientists to the discovery of the basis of all life, the cell. But it is inside the cell itself where the truly remarkable things happen. In order to understand the genome, we must first understand DNA. Shorthand for deoxyribonucleic acid, DNA is what scientists call the building blocks of all life. Inside each cell of an organism is the nucleus, and the nucleus contains the cell's chromosomes and DNA. DNA is a long string-like molecule that holds a simple but important informational code used to direct the cell to perform certain functions.
We've known for over 100 years now that there's something inside the nucleus, or the center of every cell in the human body and other cells and other organisms, even bacteria, that codes for life. The idea of DNA, deoxyribonucleic acid, has been around now for some 50 or 60 or 70 years.
The difference between you and me is actually a small number, relatively small number of bases within that strand of DNA that we both carry. All of our cells have the same, or mostly the same DNA, but between two individuals is on a remarkably small number of differences.
A genome is the entire DNA in an organism, including its genes. Genes carry information for making all the proteins required by the organism. These proteins determine how the organism will look, how well its body metabolizes food or fights infection, and sometimes even how it behaves.
The genome is essentially the sequence of all the DNA of a species, like humans, and the identification of the genes within that sequence.
Dogs and cats and rats all have genomes. That's that string of DNA that makes up all the chromosomes in the nucleus of all our cells. It codes for who we are, what we are, has all the information from the past about our parents and grandparents, and it's really the secret to coding for the proteins, coding for the machinery that create the cells and the tissues and the organs that life emerges from.
Genome's a really simple concept. It's all of the chromosomes in the nucleus of your body, of your cells. So you received one set of chromosomes for your mother and one set from your father. So it's the constituent, both sets, that comprise the genome.
The Oxford English Dictionary suggests that the term genome may be a combination of the words gene and chromosome. But scientists now know that the genome is much more than that.
How genomics started as a field of science is related to the creation of a biotechnology industry and capability called DNA sequencing. That capability to measure the genome, to sequence the DNA, is called genomics.
Genomics is the study of the genome. So it is understanding chromosome structure, it's understanding genes, how genes are aligned on the chromosome, how the chromosome fibers are packed around proteins, how all of the biology surrounding the genetic material in the nucleus of your cells behaves from a biological sense.
In 1990, the advancements in genomics and the study of genes led scientists to one of the most important research endeavors in history, the Human Genome Project. Completed in 2003, this ambitious project's goal was to identify each and every gene in human DNA.
The Human Genome Project is basically a large cataloging of all the DNA sequence of a human being, basically looking at all the chromosomes, sequencing them, and then making an index.
Every gene in your body that's responsible for how tall you are, how fast you run, what color your nails are, how fast your heart beats, every gene that's responsible for a function was identified in this project.
By first sequencing an average person and then going through and being able to find genes in that average person, we can begin to study their effect and their purpose in the body.
By identifying or sequencing each human genome, scientists hope to take an in-depth look at the function of the genes. Studying differences or mutations within the genes may reveal the causes of diseases like cancer and aid in the development of potential treatments.
The differences between you and me, my genetics, my inheritance of genes is different from yours as well. And that it certainly does predispose me for certain diseases more than you, and you for certain diseases more than me. And once we find those differences, and it sounds like a simple process, but it's a very, very complex one. Once we find those differences, we can start asking questions like why? Why do those differences make a difference in terms of the development of cancer?
Now that we have a baseline of what the genome looks like, it's much easier to look at disease states, say cancer, and find out if there have been breaks in parts of the genomes and the pieces were put back together in a mixed up sense. So you can see kind of what the underlying reasons that some of these cancers are occurring. It helps us understand downstream where the protein's malfunctioning, what are the problems.
So the hope was that if we defined what genes were responsible for what functions, we could then easily identify when something was awry. So if a normal person who was not predisposed to any diseases that we know of, such as diabetes, if that person's genome was compared with a person who was predisposed to an inheritable disease, then we could identify what part of the gene was aberrant or not functioning properly and then treat those diseases.
And it goes beyond cancer as well to other diseases. Diabetes is an area that's very active in this and looking what causes diabetes, particularly things like juvenile onset, which is a genetic disease.
But when we really look at in microscopic detail at the DNA to understand the genes, we find out that there's a lot of variation in the genes, having to do with mutations, having to do with splicing of the genes to one another, and different kinds of copying of the genes, and different kinds of imprinting from the environment and things like that, that the complexity of understanding the genome is significant. But the rewards for studying the genome to try to understand the diversity and its complexity can benefit society in understanding health and disease. It's really endless what we can do with genomics if we study it appropriately.
One of the most promising developments to come from the study of human genes and proteins has been the identification of potential new drugs for the treatment of disease. Proteomics is currently being used to analyze research and discover treatments for Alzheimer's, heart disease, and certain types of cancer.
Proteomics is the study of protein expression as it relates to human diseases and normal function. So many have proposed that by studying the human genome and the expression of those genes, we can affect human disease.
Proteomics is similar to genomics. You might notice that there's this omics at the end of genomics and proteomics. Genomics measures the genes. Proteomics measures the proteins, and by using proteomic analysis, we can understand how the proteins are changed and modified and linked together and interact so that we can understand the basic machinery of the cell. So proteomics and genomics are really quite linked together.
In order to understand proteins better, let's take another look at our DNA strand. DNA contains information to make every part of our bodies. Each of our thousands of genes codes for a specific part. RNA, a type of nucleic acid, works to transfer the information used to build proteins. But while DNA stays inside the nucleus, a type of RNA called messenger RNA copies the information in the gene and travels out into the cytoplasm. There, a protein-making machine called a ribosome will read the RNA to make a particular protein. Part of the RNA molecule codes for an amino acid. Proteins are made from amino acids. Once the amino acids are combined in a specific way with the language of DNA and RNA, a protein is created. Some proteins form structures like teeth, hair, and skin. Others can create senses by being coded to read light, sound, or flavor.
We study how those proteins are manufactured and secreted and the functions that they promote within and without the cell.
When you look at proteins, those are the things which our bodies run on. That's the chemistry that makes us happen. It's protein-protein interactions. We are nothing in one sense other than protein interactions. So in proteomics, there are vastly complex ways that we measure the amount of and number of proteins present in the body.
Proteomics can analyze what makes each human's genes different from one another on a cellular level. These differences, called single nucleotide polymorphisms, are what scientists are now studying in order to better understand disease.
Single nucleotide polymorphisms are single nucleotide changes in the genome. And when you use very sophisticated techniques, you can identify SNPs. Single nucleotide polymorphisms are also called SNPs. It's easier to say. In cancer, SNPs have been utilized to start to develop non-protein-based biomarkers to what particular type of disease, cancer you have, whether you'll be susceptible to certain treatments.
One of the untold stories in human health is that our cells know when they're supposed to die. Well, if they lose this ability, which is possible with a single nucleotide polymorphism, you have cancer.
For many years, there has been an understanding that cancers begin oftentimes because there's a mutation in a significant control protein that results in an altered cell growth, and oftentimes it is an uncontrolled cell growth. Most of the time, we have processes within the cell that are self-checking and cause the cell to go through processes of cell death, and most cancers go undetected because they arise and the safe checks and balances in our cells naturally cause the cancerous cell to die. Occasionally, you get a mutation, though, that goes undetected and it starts to grow. Then you get an accumulation of mutations in a cell, and then the cancer becomes even more difficult.
As the latest applications of proteomics and genomics begin to emerge, it seems as though a major breakthrough in disease treatment could be just beyond the horizon.
Genomics and proteomics hold a great deal of potential and have already helped us understand in fundamentally new ways many diseases. In cancer, for example, genomic and bioinformatics analysis have helped us to identify genes that are directly related to the cause of cancer, like the p53 gene or the BRCA genes from breast cancer and others.
And these are things we're starting to research and understand by using bioinformatics in collaboration with genomics and genetics, which is looking for differences in mutations between individuals, and trying to piece them together to construct the whole story of what's causing these cancers. From that, eventually, we may be able to alter or fix the mutations that are causing these in order to prevent the onset of cancer. But at a minimum right now, if we know that somebody is predisposed, whether they show it in their family history or not, if they know they're predisposed for certain types of cancer, that may suggest certain lifestyle changes they may want to take.
Type 2 diabetes was just an epidemic in this country, with more than 30 million people now suffering. So what the goal often is to understand the complex disease, understand the genetics, use proteomics to find the markers, but have that information provide us, frankly, the information about the severity of the disease, how it fits in with our family, and, where we came from, and, gives us a way to work to prevent, work to intervene early, and work to reverse the long-term chronic disease process that often faces us.
We know how to kill a cell, whether it's a cancerous cell or a normal cell. We pretty much know how to kill it. The question is, can we kill it without killing all the cells that surround it? So as we develop smarter drugs and use nanotechnology and biotechnology systems to do that, we can treat, hopefully, any cancer at any stage in any body. That would be a real nice thing.
Once the research is complete, what's next? What do researchers do with all this new information?
One of the biggest challenges in proteomics and genomics and all the other sciences that end with omics has to do with the enormous amount of data that's created. The human genome, to give you an example, has 3.5 billion base pairs of DNA. And so when we measure that with, the new sequencing devices, we get up to a terabyte or more of data. A terabyte is 1,000 gigabytes.
Bioinformatics is basically being able to take large amounts of biological information, whether it be proteins or even looking at the ecology, and being able to categorize it and sift through a lot of the data with computers and come up with commonalities. The only way it can be done is with a lot of computer power.
It's tapping into the data. It's analyzing that data with computers. It's relating that analysis to other analyses that are done with computers. That's what bioinformatics is about. So, it's clear that you really can't do genomics or proteomics without the bioinformatics and the computing to support it.
To put it simply, bioinformatics uses computers to sift through massive amounts of scientific information. It then cross-references all of this data in hopes of finding commonalities or patterns that could signify new discoveries.
We can use bioinformatics to help us determine whether we're susceptible to various cancers or other diseases, as an example. In society at large, we can use bioinformatics to help, the police figure out who did a crime. We can use bioinformatics to help measure the health effects of, exhaust coming out of cars near the freeways. All this can be informed by applying bioinformatics analysis.
A lot of cancers, once they're removed surgically, the next step is to decide what next. We just wait and watch. Do we give treatment to try and prevent the recurrence of cancer? That's a more difficult question than it might appear, because a lot of cancer treatments are very dire, very difficult. So you have this difficult decision to make. So the tests we're working on, analyzing this data, are designed to add additional information to help to make these decisions.
So, for example, if I was unfortunate enough to have a liver cancer, my cancer would be unique to me, and each patient's would be unique to them. However, you can apply the tools of bioinformatics to examine expression of many genes across all of the genome and compare multiple patients. And you begin to get information. And so bioinformatics is really changing the way in which cancers are being treated today.
We know that Alzheimer's disease tends to run in families. and so many diseases run in families. But, it just hasn't turned out to be that simple to understand how the genes are passed through and what the effect of the environment is. And bioinformatics analysis and genomics and proteomics analysis are helping us to unravel, what's going on in the progression of Alzheimer's disease. And Alzheimer's, like many diseases like diabetes and other diseases, are what we call complex diseases. They're not just caused by one gene. They're caused by the combination of many genes interacting with the environment. And so we are using computers and modeling and analysis to understand what we call complex diseases that have many genetic contributions.
The crucial element is to take all this data and extract something from it, and that's what the computational part of bioinformatics is. That's why it's such a fascinating computer problem, to take this huge amount of data, which is critically important, and extract the key features with it, and then to work with the scientists to say, here's what we found, can we put this into context? Does that lead us to other questions?
Everybody tries to look for their own answer, but with a computer, it's purely numbers. It's purely ones and zeros once it's all put in and sifted through, and you don't have the bias. So you can find unknown correlations that you wouldn't have thought to look for if you were just putting pen and pencil to paper and trying to sift out through all these different charts. So it has the potential to unlock quite a few unknown pathways we hadn't seen before.
Across the globe, doctors, scientists, and researchers have dedicated their practice to the study of genomics, proteomics, and bioinformatics. Through their studies, we are able to see beyond the limits of our own physicality to the very structure of life itself. And one day, they may make the discovery that changes everything.
The 20th century was all about physics and the nuclear age. The 21st century is all about biology, medicine, life sciences, bioinformatics, genomics, proteomics.
Whether we will ever have the ability to go in and fix a gene ever is a long time. I suspect the answer is yes, we will ever have the ability to do that. How soon that is the question.
It's an interesting fact that when the human genome was sequenced, everybody said, okay, great, so tomorrow we're going to have new drugs that'll cure all these diseases. And there was this period where they said, well, where are they? We're 5 years on, 10 years on, whatever, and where are we? But people have to remember that it takes 10, 20 years to go from the concept of a possible drug to a drug that can be safely sold to people. And so we are just now creeping up on that time frame where we'll start to see the benefits of it.
It's an ever-changing field, and that's what's so exciting about it, that you're finding out new things. Being a PhD, one of the things I've learned is that it's more of how much you don't know. And that's the enormity of it, is how much we don't know, and we're all working to understand even more.